will be more delightful and memorable. Now I request Dr. N. Raghavendra Naveen, coordinator of this program, to handle the session and introduce the speakers to the delegates. Over to you, Naveen.
Thank you, ma'am. Very good morning and warm welcome to the respected speakers, faculty, and the participants. I, Dr. N. Raghavendra Naveen, working as Associate Professor in the Department of Pharmaceutics. So it's my privilege to introduce today's speaker, so Dr. Nilesh Kumar Mehra sir. So sir is currently working as Assistant Professor. So in National Institute of Pharmaceutical Education and Research, Hyderabad. So prior joining, he worked as a manager of product development.
Sir, unmute your mic, sir.
So with a professional of 12 years of experience in the parenteral formulation development in the pharmaceutical sciences. So has completed his postdoctoral experience. So from Texas A&M Health Sciences, USA, and also worked on ophthalmic and injectable product, including nanoparticles, liposome products. So he is expert of process optimization, design optimization, so using DOE and QBD studies, and research updates in the field of drug delivery. So he is also the editorial board member of current drug delivery journal and many more reputed journals from elsewhere, Springer. And till date, so his research group has published more than 60 international publications. So with more than 2,000 citations and a H index of 29. So with a cumulative impact factor of 350 in a high reputed journals, and more than 20 book chapters in a CRC Press elsewhere and a Springer. So were published by so Dr. Mehra sir, and sir also received a grant of around 15 lakhs and 3 books edited and published. So along with that, 16 book chapters were published. So he received awards like Team Award from Centris Pharma and the International Young Scientist Award and the Young Scientist Award from DBT. And sir also currently as a reviewer and editorial member for various scientific journals like Journal of Control Release, Journal of Drug Delivery Science and Technology, Biomaterials, Drug Discovery Today. And I request Nilesh Kumar Mehra sir to share his valuable experience with the participants through the topic entitled QBD approaches in the product development. Thank you, sir. Over to you.
Thank you, Naveen. Is I am audible for?
Yes, sir. It's audible, sir.
Okay. So thank you for a nice and brief introduction part of about me, my educational background and research areas, which I am doing from my best efforts. So now, shall I continue for the share screen?
Yes, sir. You can continue with the shares.
Yeah. Thank you. So I'm sharing the screen.
Okay,
now. So please just confirm that it's visible.
Yes, sir. It's visible, sir. Yes, we can go with the.
Now I have slideshow. I hope it is in the slideshow mode.
Yes, it's clearly visible, sir. You can continue the session.
Okay.
Thank you,
so thank you. I would like to thanks to all faculties and AICT's faculty from the Annamacharya College of Pharmacy to give the chance to speak about and share my knowledge on the QBD in the product development, specific for the complex product development. So I am so happy for delivering this lecture. So my topic is on quality by design approach in the complex product development. So in which I would we like to talk about this, what does it mean by the product, and but particularly we need to be focused on the complexity and why applying of this QBD and the other approaches. So many approaches had been come in the QBD, so that we can be elaborate and might be get the. I would like to share my experience in this QBD. So before I start with that, I just want to say that this presentation is not the part of not represent any views or any from the neighbors views or the regulatory or policies part. And I have taken the information from the various sources, including the scientific journals, the different peer reviews, published articles, and the book chapters and the books, and I have taken some data from the regulatory guidance from the different regulatory agencies, including USFDA, WHO, EMEA, or some other also. And rest of the part I have collected from the sources that anyhow they are the scientific one, and I have utilized the Google platform for collecting this data. So first I would like to tell about the complex product. What does it mean by the complex product development? So that we can understand about what does it mean by the complexity. So as we know about that, we have the two terminology, that is the simple versus complex. Previously, in particularly in our India, we are working on some simple or non-complex products. What does it mean by that? The simple, in that we usually utilizing of like a 1 or 2 unit operation. That is, the manufacturing process is not longer. We use for example, if we want to prepare the one solution, like you have seen here on on tears or the refresh tears, or the any simple solution. So they might be involved only the lesser unit operation. Lesser unit operation, that means the manufacturing line that is available in the plant for manufacturing plant. So that having only like a 1 area, 2 area, or 3 area maximum for the manufacturing, and then it would be shifted into the packaging like that. So we are not utilizing so many instruments. We prepare in the one manufacturing tank that is made by the SS316. So and after that we can filter it and transfer it to in the sterile area, inject sterile area, and after that we again mix it and make up the volume, and then transfer into the packaging area with the use of filter. That is usually in case of sterile, we usually consider 0.2 micron filter. So that is the very easy process, and over there we consider some critical parameter, process parameter. That is only I can say only one parameter or two parameter that we can say like a process parameter. Then critical quality attributes that we can say only 1 or 2 like that. So this they are not so much complexity on the process is not involved in the more complex, and their assay parameters or the other characterization parameter is not a lot of factor. However, in the another side, you can see the complex product. The complex product, you can see here with the example of nowadays we are talking about the nanotechnology as well. So like a liposome, nanoparticle. Similarly, you can see of this complex product that in an ophthalmic adjunct, that is the one brinzolamide ophthalmic suspension 1%. So it is come in the complex. Why we consider it is in a complex product? Why? Because it involves multiple unit operation. It involves the change in this specific requirement, this equipment. So in that we usually involve a milling technology, in which bead mills or the microfluidics technique we involve. So these equipment, they are the critical equipment particularly dedicated for these complex products. So that's why what happened, we are involving with the multiple unit operation. That means the process we have to be, the process is very complex, and we are utilizing of a complex instruments or the complex this equipment, dedicated equipment that we can say it. So that is the one example for this. Similarly, you can see also one other ophthalmic formulation, that is the Durajol ophthalmic emulsion 0.05. It is a nano emulsion. The size is, the mean average particle size is around 130 nanometer. So in specific for this Durajol, the microfluidics technology is work on that. So this is the innovative one. If in India, we are working on some generic formulation like this, similar with this Azopt and the Durajol. So we need to have all process unit operation aligned with this Durajol, and all this critical process parameter, critical qualitative, that should be meet as per this reference product, that is the Durajol. Similarly, apart from that, some this you can see some conjugate products or the lipidic based product. So over that we also involve a multiple unit process operation. So this is the difference between the simple versus complex, that is clear crystal for that. And now the scenario, we as a pharmacist or we like a formulator researcher, we are entered into the complex product development, not only in the generic, even though for the innovative product development, we have been working on this complexity. So, and similarly, we are entering into the biological product as well as. So this is the scenario of or genre of the complex product development. So this is the that one. So that is also for more elaborative one for this complex product development like that. So what I already spoke about a lot of things, but here like that. What is involved in the complex product development, like complex APIs? What does it mean by the complex API? Like if you are taking the peptides, low molecular weight heparin, for example, or any with the help of some natural synthetic polymers. So they are very critical, like for I'm taking the example of complex API. Like for this benzolamide, we need to take the API, and because how this is a suspension product, so we need to be take for the only sterile API because the size of the API is much higher. And after if we complete it, so after that we can't go for the terminal sterilization with the help of filtration. So we need to be take the sterile API. This is the one challenging we can face. So for handling of the sterile API, their sterility, their weighing issues. So a lot of these challenging technical challenge we face into, for executing the batch into the plant. And this, if we are unable, we are face failure in this these parameter. So our product will not be meet this desirable specification that we have set into for the quality terms. Similarly, a complex formulation for that liposome and this colloidal or suspension nanoemulsion. So we are using this multiple unit operation to make these all parameters have been in our within a range. So that is the one. Second one, and the complex dosage form for like example of some intravitreal injection that we are preparing. So that is the this is the communal more complexity development of the dosage form. Complex route of delivery, that is also like this suspension if we fall gel or ophthalmic gel also. Complex drug device combination, nowadays you can have seen like that so many medical device product that is based on the drug device drug device combination. So that is also available in the our market, and every day we are working on some more advanced technology like on this drug device combination based, like for example intravitreal injection into the eye. So the drug should be delivered into the vitreous region of the eyes, and even look like the some delivering of the posterior drug into the posterior segment of the eye, like advanced technology in the contact lens, needle-free injection, auto-injector. So these all are coming into the complex drug device combination that is also in the new era for complex product development. For in the generic as well, if we are talk about the generic, and we need to be make that, so we have to show the equivalence studies, pharmaceutical equivalence as well as bioequivalence study to for and we need to be compare our product with to the innovator one. So then we need to be perform the in vitro bioequivalence, and before that, before this bioequivalence, for this complex product development on, we need to be match the Q1, Q2, and Q3 similarity, sameness. And after that, that is the also one very tough job for getting the approval of Q1 and Q2 from to the regulatory agencies. So we need to be we need to be get the approval of this formula for complex product development once we have submit to the, like for example FDA. So they will evaluate it within and respond within 60 days, 2 months, that either our Q1 Q2 has been approved like in line with the innovator or not. So they will like, so for that, you know, for this Q1 Q2, because we are we are adding lot of additives or excipients. So like for example of EDTA, if we are using as a like antioxidant chelating agent, so we need to show the like EDTA, which form is that, is a EDTA dihydrate, monohydrate, and like this is the for example one. Similarly, APIs, if we are so that is the anhydrous or the having like crystalline kind of that, or some other excipient that is the like a combination of phosphate buffer, dibasic, monobasic hydrogen. So you know, for getting of the Q1 is because it is available in the label of the FDA. But for finding of the Q2 value, that is the quantitative one. So that is the difficult for analytical peoples. So they need to find the Q2 value, that is the quantitative one, from the reference product. And we based on with the help of analytical formulation experience, and then this obtained value, we need to set up the Q1 Q2 formula, and we need to be submitted. So for first we need to be obtain the Q1 Q2 upload, then only we can execute of R&D activity, and then follow for the manufacturing plant for the feasibility and pre-exhibit or exhibit batch, and then validation batch. So it's a very complex. That's why we consider what does it mean by the complex product development and like that. So before this discussion about the QVD, but because this QVD approach, why this regulatory agencies has been entry into this for product development, because due to this complexity. So later on, a lot of challenges we face as a manufacturer for company side, and there is a lead for the failure also. Then only after this Q1, Q2, and Q3, that is the physical chemical examiner. This is just recently introduced one more terminology. Then a release in vitro release techniques, and then if ophthalmic, if permission studies, or in case of we need to show the in vitro bioequivalence studies. So reference product, I might know everyone like that. We understand about the reference, either somehow some regulatory guidance they consider like reference listed RLD product, somehow they consider reference product, somehow like innovator. So depending upon to that, each country is having their own regulatory guideline. So like that. So we need to be show our equivalence product to this reference listed product, RLD product. And in case of USFDA, this is available, these all information labels, their quantitative value, qualitative values, and the chemistry parts, and are available onto the USFDA website. For you can get download the labels, you can download the chemistry review, you can download the pharmacology review for understanding of this product that is from the innovator side. So that is it, we can get it. So for that, in vitro and in vivo characterization we need to be performed, and then we show the bio equivalence with respect to the innovator reference product. That I also speak about this combination product. Now it's very important in nowadays. So that is like one combination of drug and device combination. So in that we can say for example of needle free injection. So in that you are in a nanotechnology, we are using a nanosphere, this PLG based kind of microsphere nanosphere based formulation. So in that we prepare, and then we can why after that you see, once we prepare the microsphere or nanosphere, then we need to be lyophilization. So for setting up lyophilization, their cycle, their temperature, time, we need to work a lot. And after that, we have been put into the device. So that's why it is come in the drug and device combination. So first we can say a single entity combination product that has been comprised of the 2 or more regulated components. And after that, we can have been packed into the this device. So that is the co-packed combination product. And after that, we say that cross-labeling of the device or the combination product, that is we can do. So this is about that drug product, drug device combination in that, for example, nano needle or needle-free injection kind of that. So you can download the drug product combination guidance as per the USFDA from this link that I have collected the data and read it about this combination drug product. For this also, drug device combination product, in that the compliance of the product license 21 CFR part 4. You might know about the CFR. It's code for the federal regulation is having the subpart 50, and then each subpart having the particular subcategorized form. And similarly, medical device directive for pharmaceutical in case either medical device or medicinal product. Now coming to the QVD, what does it mean by the QVD, and why, how we can be implement into the during to the product development. So this QBD concept is, so in that we can say like that the quality cannot be tested into the product, but we can be built into the during to the product development. So that's why the concept of this QBD has been arrived into the regulatory guidance for getting the as per the compliance with the international ICH guideline and specific Q8 guideline. So this is like a very systematic approach and to understand about the and also to manage of a product life cycle. So with the employment of this QBD, because this is systematic, holistic, and like we can say also proactive approach. So for managing of this entire product life cycle. So with the why we can understand with the help of what we are formulating and what is the predefined objective with us, what that we want to be understand, and so that our product has been to be go in a very smooth way, so that the product there is less chance for to the product failure. So with and how we can get it, so we need to be emphasize more on to the product understanding, like for in the including of generic as well as innovator both. So the product understanding is very much very required. And after that, what kind of the APIs, active pharmaceutical ingredient, we are in covered, either this the sterile one or the non-sterile one. Likewise, and all APIs, those are regulatory approved. They have their DMF drug master file is available. We can download, we can see into the website. This regulatory website is the product or this API has been the DMF is available on that. In the DMF API, this manufacturer provide all details related to the API, including the stability study as well as stability commitment. So for that one, product and understanding, and second one like a process understanding. How if I am the formulator, I know about this complexity based on my knowledge, based on my experience and my mind. So we can understand about the process, how I can be execute this product, what kind of the process is involved. So that kind of understanding is required. And similarly, like if I know the process, I know the understanding about the process, then we need to be identify and mark it where we can control it. So that is how the emphasis more emphasizing on the product and process understanding and the process control. These are like having like a that's why it is called, you can see here on the first scientist systematic, holistic and proactive approach for managing. And here also emphasizing more on the product and process understanding and the process control. So ultimately what we want to be like the product should be easily available. We can manufacture it, increasing the process and product understanding, and so that our the risk associated with this product to the patient is we can minimize, or we can say like a minimal risk for that. Sorry. So this is the like. So with the understanding of, with the help of the scientific approaches, or with the help of the, that is the one QRM, quality risk management. So on the process understanding these all, so that we can do for a successful product development, particularly complex. So this QBD we can't say is one like just introducing or a new one. It is like a new. It's not a new. It is having the like, we understand about, we already implemented that one, but we are not applying some this systematic approach. So that one, that's why. And with like a DOE, design of experiment, we know it, and the designer space, we know it about that. So you can see here also for this the key aspects or elements of for this QBD approach. So this QBD is also similar with to the PDCA, that is the iterative approach, that plan, do, act and check, plan, do, check and act, that one. So that we need to focus on to the risk based approaches. We need to be always ready, and we always employ ready and also encouraging for the continuous improvement of the process and the product understanding, and for the this minimizing of the error. So this is the QBD is a process defined with the help of documentation requirements. So why it is like as I told that why it is required. So in the generic pharmaceutical industry business, the regulatory perspective point, if we talk about that, what previously people are thought, they thought that we need to develop the product, we need to file it, and then we'll see later. So that is the file product first, learn later. But this is not case into the nowadays for the complex, because if one product has been failed, it create a lot of this business loss to the product, this manufacturing pharmaceutical companies or onto the formulator as well. Second, like major amendment during to the review process. If we file the product, they will regulatory agency will be reviewed, and then we will say that amendment. So that exhibit batch stability failure. Definitely, if we are not working properly, so there is might chance for the exhibit batch failure mode. Formulation, we need to rework, we need to relook, and then we again go for the exhibit. So it is taking off a longer time for development of the product as well. So this is the failure for the research of the formulator. And then regulatory person will say that it's a failure for the formulator. They are not able to develop of this one product and process. They are not aligned in that case. So for, until unless what happened, the product will not be approved, and then it not be marketed. Or in the case if the product has been approved, some changes has been like a post approval changes. So that is also for we need to file it again with the help of regulatory. So with the help of this QBD, because this is one, and so what we can get it, we can get the avoiding of the regulatory compliance problem. So our product is continuously selling into the market, and more efficiency and the cost effective terms for the safe for the industry point, and then we can ensure the high level of the product quality, their improvement of that one, so that also we can monitor, we can track it during to the product development and after like a product development as well. Minimizing of or eliminating of the this other compliance section, that like a market survey or like that, eliminating up to the exhibit batch failure, validation batch failure. And also if we work on that right track, so we chance for the minimizing of deviation, costly investigation like that. So a lot of benefits we can get, and until this we can get that one product should be available onto the market for the patients health care. So this is the key aspects you can see here on the right side, that is the documents process related to that. So target product profile, TPP quality, particularly early product development that we can execute into the lab R&D. Risk assessment, like once we have to identify the qualities or their target product profile associated with to the complex product development. That's why I spoke about in the initial slide, what does it mean by the simple and complex process development. Then we need to implement the control strategy. There also the risk has been associated. After that, we develop and then transfer the product to the plant manufacturing plant, so that we collect technology transfer once it has been executed. So after technology transfer, once the process risk assessment we need to be executed, over that we can work on the risk assessment, that one, and then we need to execute the batches, and then that would be like we can say like a process verification or for the continuous process verification. Over that also risk has been associated. So this is the one. So what QBD does, so that is so this QBD is related with to the formulation and manufacturing process variables, CMS of API, excipients, or the critical process parameter, as well as the product characteristics, that is the QTPP. QBD principle, increasing the product understanding and process knowledge, that I will cover. So I don't want to again see this one. Yeah. So for this it is a very multi-dimensional. Just wait this. Yeah. Now coming like this design space is also is. So this is a multi-dimensional combination and interaction of like input variables, input that variables that is related to the APIs, and this is related to the materials, excipient with that we are critical material attributes, and also for the CPP, that is the critical process parameter associated with to the manufacturing. So in that, we need to be out like this design space, which one is like out of within our design space, which one is like out of the design space, that we need to be identifying. So like you can see, like the risk-based concept and the principle by involving of the Q8, Q9, and Q10 and Q11. So that is a somehow like related bit to this knowledge transfer, knowledge experience, this experiments, and the risk assessment as well as the product development. So pharmaceutical product development is complete, including in the Q8. However, the risk management and the quality QRM, quality risk management, for structuring of the process thinking, that we have covered in the Q9 as per the guidance, and Q11 is for the this quality system like also. The new quality paradigm, if we talk about that, you can see here also this new quality paradigm is more emphasizing onto the product understanding, product testing, inspection, and also utilizing up to the modern science throughout the product life cycle, even though by including of this QRM, quality risk management. So this quality risk management is very, we can say like a key enabler throughout the product life cycle. So with the employing of this product or process qualification system, be it for knowledge management or integrated approach, we can be develop the product and to the for both, and so that we can save the time for the industry as well as for regulatory. So you can see also this is the one new product from product profile to CQS, risk assessment, design space, control strategy, or continuous improvement. You can see here also how this is, the all are associated with each one. Quality target product profile, SOPs, process analytical technology, PAT, RT, and it is coming to the design space. And then you can here also you can see how similarly we can parallelly we can work for the identifying the CQS, QR, identifying the CMS, CPP, and the PAT technology for the product development as well as for the analytical tools. So these all we can combine it, and then we can say that we can control, and so over there we control strategy has been used. So steps, this is not. Steps involve the quality by the design product. So development of the new pharmaceutical product. So that this already have been executed in the R&D, and then we need to go for the pilot batch, and then exhibit or 3 exhibit batches required for regulatory filing. So commercial batches is then scale up the and like validation batch, pre and post validation, and then we can submit for the slide. So in manufacturing, we need to apply for the design space and the process analytical technology for the analytical tools that we can implement it. Now even like HPLC and LCMS, we are using this kind of approach for that, and real-time QC that we can implement in the manufacturing process. Control strategy, as I spoke about that, based on this risk that we are initially scrutinized and made it, and then we need to be rectify the risk, and then we need to be so that the risk should be minimal, and after that continuous improvement so that the product performance should be more. So like this, you can see also how has industry responding this, the different regulatories. It is like somehow it is very confusing for us, like which one is like come into this QBD. So like pharmaceutical quality risk management is that I say that it is associated with the ICS Q9. However, the product development is associated with the Q8. So that one is also similarly, like if risk management we need to be there. So in that we need to be take care of for the product and process understanding, design, control strategy. So that you can see here also in the at the bottom and the left, this right side. So criticality for that is associated with the product understanding. So we understand about the product, we understand about the APIs, their structure, this based on the knowledge, and we can collect from the information from the DMF. And after that, we can correlate this CQS, quality target product profile, or the CMAs. And after this understanding, we need to move on the process understanding. Then we can need to be completely exhaustively work on to the operation, unit operation, how many and the materials that we are using, like for example, polymer we are using. So we need to work on to like a polymer, their viscosities, and what kind of the viscosity we can use, like low, medium, and high grade up, and how they impacting onto the product, how they will be impacting onto the within process, and how they will impacting onto the final product. So based on this understand, we can implement the control strategy so that over there we can reduce or minimize the risk associated for the product development. This I spoke about the quality key aspects that required. QTPP, CQS, risk assessment that we initially perform, critical material attributes, CPP, design and implement of the control strategy, then product life cycle management with to continuous improvement. So now I have so many times I said that QTPP, CQS, CMS, and CPP likewise. So I want to be discuss about that. What does it mean by that? So the quality target product profile QTPP. It is a very necessary element for the product development. So in this QTPP, we understand about the qualities characteristic up to the product, complete product, finished pharmaceutical drug product. So like including of sterility, purities. So anyhow, like we can say like this, once we understand about the finished product development, that we can plan with like a once initial like till to the end of the product and approval. So that is the like in case of generic formulation. So that is one advantage with us. We have one reference product, so we can work on according to them. But for like innovator one, we need to be think about proactively and for the end of the products, a finished product. So that one. So this is the one you can see for starting of the product. So you have the knowledge, you have experience, you have a expertise in that case. So you can utilize that one. So for understanding of the quality target product profile, then you need to be work on like what does it CQS involve in the quality target product profile, so that we need to be work on that one. So for example, the quality target product profile, like for that one tablet one, you can see what does it quality target. So whenever we talk about the quality target product profile, so like we need to be like once it's like a tablet, what kind of the dosage form, solid dosage form, what kind of the route we need to be give it to the, yeah, definitely oral one. What is the disintegration time is that, what is the physiochemical properties, that what is the stability of this drug product is associated, we can show it. So like this, for example of the process of product development of the semi-solid in case, so we need to be this is the complete cycle of understanding about the semi in case of semi-solid by including of CMS of the APIs, excipients. Similarly, for the process understanding with the help of critical process parameter that we need to be identified, and with the help of CQS of the semi-solid product. If like in a nanoparticle, this size is concerned, so we need to work on the particle size, volume of it. Semi-solid in case, so we need to be CQS is the this rheological profile. So this is like, what does it mean? They need, they initially we can work, then after that we need, these data have been, that would be helpful for setting up to the specification limits. For example of QTP that I showed, that the semi-solid that I already spoke about that. So this is dosage form, route of administration, dosage strength, what and stability, viscosity, content uniformity, microbial content, like that. So these are all quality target product profile elements in that case, and this kind of we need to be work on. And after this we need to be add this data, this record into the product development report. So we need to this QTPP element, the target, and then is this involved on the sequence and what the justification. So this is kind of the one format, every manufacturer having the pharmaceutical company having their own SOPs, so that we need to be compliance. This one also. For example, in case of tablet that I also dosage form tablet, what the strength that we need to do. Assay is specific impurities, like this any other parameter that come into the QTPP elements. And yeah, QTPP and the now nano formulation, like for example lipid nano system, you can see. So that we need to we identify the QTPP, like for their administration, their pharmaceutical dosage form, their type kind of the lipid nanosystem is that, what is the final product is, their quality, safety, and efficacy is that, and what does the CQS is involved with that lipid nanosystem. That is a different one. I will also speak in that. And then CPPs and the how they are associated with the risk assessment. So risk assessment initially we can do it, and then there is a chance for minimal risk. Now another this one case studies for this QVD based. You can see the QVD guided early pharmaceutical development study, production of the lipid nanoparticle with the utilizing the HPS techniques, high pressure homogenization for skin cancer treatment. So this is the one you can more understand about the quality target product profile for finished hydrogel formulation. You can see how they identify the QTP. So route of administration, any other methods, no delivery system with they because they prepare the lipid nanoparticle. So that one dosage form what we are preparing hydrogel, that is we need to be mentioned what kind of the therapeutic effect or therapeutic indication that we wanted. What is the viscosity, what the release profile, occlusability like that, and stability or like that also. Similarly, the CQS I am just giving because this is case study. So CQS, that is the critical qualitative related for physicochemical characterization of the product. Like in case of this hydrogel, like because they are preparing lipid nanoparticle, the particle size is major concern. So we can say this is the CQS. In that they prepare the specification for the window of the nanoparticles size range. Polydispersion index is also playing very important role in that. So we kept into the CQS, zeta potential, encapsulation efficiency, percent drug release. So that would be come into the critically quality attribute. And other parameter that we discuss in the upper one, that is having the quality target product profile. So like quality, what does is the components that I am giving a more understanding for that. So the components that we want to discuss about that, they are related with to their safety, efficacy, identity, purity, and the potential. Like what is it, the critical and non-critical component example for of any product, like critical. So firstly, we consider like assay of the API. Assay of the API is a very critical. If it is get failure, the product we need to be considered in a failure mode. Similarly, like content uniformity is that non-critical, we consider like a description. What is the physical appearance or description of this product? Like if we have a clear, colorless, transparent formulation, so that is like it will be change in the stability or this like we can consider like a non-critical, like that one. But it's not like that. Clear should be like in a red one or a brown one after half an hour. And similar like a fixed and variable component, fixed elements must be present, like in dosage form that we can't change it, strength that we can't change it. So these are like a fixed and variables. In variable elements, we have range of some acceptance range, like for example, assay. So assay of like suspension, for example. So we need to be set like a lower limit as well as the higher limit. 100% assay is there. So we need to be of work on the different batches. And based on that, we need to be set the limit for a specification like plus minus 5%. So 95% is the lowest one and 105% is the higher one. So how it would be changed, because then we know about that change in the volume is changing like a error or some error. So that would be if it might be go from higher side, it might go to the lower side. But beyond that one or lower that one, we need to be considered as a failure mode. So that is we have considered also like a fixed as well as of the variable elements with having the acceptable range. So this QTP, for example, one more emulsion that I also like dosage form, route of administration, strength, physical or chemical properties, storage, sterility, shelf life that we consider. Like you can see all the Durajol and the Restasis. These are the ophthalmic products having by the ophthalmic emulsion one is that. And this QTP will lead to the critical quality of, if once we understand about the product and the this quality target product profile, so then we lead to the understand about the CQS. So this CQS we know, and then based on this, because this QTPP, once we understand what is the QTPP, what form of the basis for the QTPP, and what parameter we need to be defined is that, and then we lead to the understand about the CQS. And for like in a generic case, what is the basis for this QTPP? That we need to follow up the reference listed drug products and their level or the applicable regulatory guidelines, and then we need to be identify and we need to be make the quality target product profile is that. Second one, like it lead toward to the CQS. Yeah, once we find out the QTPP profile, then out of like you can see like a particle size, that is the one example for the CQS understand. So this is like we can say also the CQS are the subset of the QTPP, and or I can say like that the CQS are come into the QTPP, so that it include of the parameter that have been likely to change based on upon the like variation into the input material, raw material, excipients, or the process has been changed. So like that, like for example, identification test. So this is not we can consider like in a CQS. Like for example, assays of the API, like content of uniformities, particle size, PDI. That is the example of the CQS. So for well understand, and this CQS, once we said this doc and it has been documented, we need to be monitored throughout this product development till to the even though after the approval also or the product has been available in the market, those batches we executing, we need to be always perform this CQS and report it. So this CQS, they are very important, critical, because we consider the CQS critical one. So they, because once this CQS have all the CQS which we identified meet into the within a specific, so that we can say also the product has been remained within its or specific the safety and effective levels. So that's why we know. And the, for example of this QTPP left side you can see, and after that will be converted into CQS. So like from physical attributes you can pick up the critical quality attributes like assay, particle size, globules, impurities, content uniformity. Like these are not limited, it's assay, impurity, and content uniformity. So once we have a QTPP profile, we can take up what of the CQS is associated with that product. So this is a completely entire teamwork, collaborative work with the regulatories, quality assurance, and like that. So this is QTPP and CQS related for involvement for the specific specification development, like within in process specification, finished product specification. So that we need to be helpful for this acceptance criteria and for the leading to the for development of a good safe and effective product development. So you can see also like the desired target for development work component of QTP we can say, and then it correlate with for the CQS, and then we establish the set of the criteria for acceptance of the specification, so that we always taken off leading to the product development or the product development. QBD is iterative process. Yeah, just wait. Sorry for that. So we can say that like the QBD is a very I plan to check and act PDC technique. So it having the multiple feedback modes. You can see also how they are each interlinked with to each other. That I might be, if I explain very well, so then you might be understand about that. The like quality target product profile, if we said, and it would be once we say go for the initial development, this quality target product file, they are impacting onto the development, early phase development, that is the in R&D level. So they are interlinked with each other. So for complete product life cycles, we can employ this QBD approach and on any stages. So this is brief about like that understanding about the where we can apply this QBD approach into their product option. So this, in that case, we can minimize the risk assessment or risk management in the as per the control strategy. And you can see also in the left side process development, how this process development is related with like a technology transfer or like a process verification techniques or for improvement. These are all interlinked. Once it is like a if process risk assessment is related to control strategy. So in that, if we work systematically in a initial way or in a proactively for to get the product finished product development, so that is a we can get the definitely safe and effective or product without any failure. And here we can say also like the risk has been associated in each part, either the QTPP, CQS, and that. So we need to work on the systematic holistic approach for implementing of the controlling of the risk associated. CMS critical material attribute that this is a I think everyone knows about the materials or the any raw materials excipient intermediate that we are incorporating into the our product. So like physicochemical and biological properties related with to the materials that we can cover into the critical material attributes. Like for example, one polymers we are using. So the polymers we know about the physicochemical, biological, microbiological profile. So we need to understand of the polymer that one. For example, carbopol, if we are using. So it is available. What does the color, shape, size, and solubilities of this carbomer? What is the chemical test we can perform to say that assay? Definitely, the vendor has been supplying the certificate of analysis over there. They say us to the detail about the specification, but we need to again validate the assays or the other characteristics that cover to the physicochemical, biological like that one. So that is the one we need to work of all received excipient. We have to be subjected first for the testing as per their COAs, certificate of analysis that supply from to the supplier. Similarly, like the process understand critical process parameter. So this, as I shown that this is the subset. This is also set for the understanding about the critical process parameter. So, like in complexity, we need to be identify. We need to be mark. These are the process parameter which are very critical. If we are not working on that, if we are not focused on particular that one, so it would be changing to the our product. It's changing all the desirable characteristics of the product. For example, of this you can see in process control of process like this RPM, temperature, pH, conductivity, flow, time, moisture content, loss under. I'm saying like one example of pH. Like in case of the polymer, this carbopol particularly, it having the particular neutralization capacity. Once we titrate it, we add it, we meant we definitely we prepare the dispersion of polymer, and then we need to be maintain the pH of the product. In that, what happened in that case, it impacting on the once we add in the NaOH, sodium hydroxide, either it's a 5 normal, 2 normal, 1 normal, that we need to be also work on that. Either we need to be 5 normal, 2 normal, 1 normal, 0.5 normal, like by that. So once, like in case of this, I face this experience, sharing experience. So the pH is in when we are like 5.5, if we are increasing up to 7.5. So the range is 7.3 to 7.7 we said, and so from 5.8 to going by addition of the pH, and it is like carbomer and polymer, so it make of a gelling kind of. So there is a lot of problem into the string. So we once we added the pH, this sodium hydroxide solution to maintain the pH, it sometimes it not going to the bottom, sometimes it stuck into the upper layer or middle layer, it will not go reach to the down level. So that ultimately the sodium hydroxide is not mixing in that one. In the, even though like with the help of high speed or this bottom up this apparatus, this is high equipment. But after that, if we added 1 ml and suddenly the pH has been changed. The pH is reaching like a 12. So in that, this is the critical process parameter we need to that, and we need to be check the CQS of pH also. So this is the correlation of the CPPs and CQSL. So in that case, if the pH is going beyond the 12, the R specification is 7.5 or 7.3, 7. So we need to be stop this batch right now. So those kind of these all approaches has been we already work on in the initial early phase development, so that these kind of the problems will not face into the manufacturing site or premise. So this is all you can also understand about the correlation interlinking between to the CPP, CQS or CMS. You can see also. So like this is that I given the one example, gave you one example, CMS input material, and like CPPs like this string and this how much speed we need to be maintained, for what time. So that is involvement of the CPPS one, that is the one unit operation like one. And the material attributes that I speak about, if we are using the high viscosity, so there is a chance for that this ester will not be moved because due to the high content. If we using the medium grade viscosity or low medium grade, so that probably will not achieve the final viscosity into the finished product, so that we can work it. So how they are linking that, that's I want to tell to you guys. So what factors affect drug products? CQS, CMS, and this one. And after that, like we added one more materials. So that is also, so like one and after that, this is the one. So 2-1, and after this unit operation one has been completed, then we need to be this polymer solution, we need dispersion, we need to be transferred into the other in the second sterile units. So over there, high pressure homogenization is there. Over there, APIs, slurry is there. So that once we transfer from unit operation 1 to the second unit operation, and then transfer this API slurry and then mix it. So over there, critical process parameter related to the homogenization and then mixing that one. So a lot of this unit operation has been involved in this product development. So that's why I spoke that this is the complexity of the product development. So this QVD would be helpful for initial level, and then we documented and we minimize the risk all associated with to the product. You can see also this is the one kind of that we involve in the risk assessment initially, so we can cover it. We employ use our knowledge. Like you can see drug product sequence we identify, like physical attribute, for example size, assay, LOD, water content, drug release, and how these are related with to the drug substance. Drug substance, that means like APIs. The APIs property, the API sequence properties like solid state form, either the crystalline low, hygroscopicity, what is the particle size of that, what is the span value is that, span value the D90 minus D50 upon D10 is that, or the D50, D90, D50 value is that, what is the polymorphism nature is that, process impurities have been associated with this APIs, or what is the chemical commitment or chemical stability is there. So you can see also the drug product, like for example assay. Assay is related somehow with the particles, nature of this product. If they are hygroscopic, during to the dispensing, they might be stick into that during this bend, or during to the transfer of that API into the tank, it might be loose. So it like that. So here we can mark it, and this particle like this with the marking with the low, medium, and high classes. So now involve like a control strategy. It also, so if with the help of this control strategies, we can like this as per the ICH Q8, and a plan set of the control derived from to the current product and process understanding that ensuring of the process performance and the quality of the product. So this control strategy including the following elements, like understanding about the drug substance, APIs, excipient, or the container closure that might be involved to the products that we can use in the finished product. Process operating condition for the equipment, any process in that control, we can say that in process control, or the specification of the finished product that we need to be set for the within the acceptable range. And even also like if we can work on the each unit operation, and then we can have to decide or the set the specification in process and like that. So with this assessment is very important for that. So we need to be initially assess the risk, and then we identify who have whatever the risk has been come in the high range, high side. We need to be work on that, and we need to be minimized in a and come into the low. So that one. So why risk assessment in the product development has been involved to identify the relative risk level at the beginning of the product development, to prioritize the limited product resource or the documentation, to minimizing of this uh, error or risk, as well as for like so that we can take off the risk. It should not be come into the skill up or the technology transfer or the execution time. So that one, that I already speak about the way that this has been an associate with to the this finished product development, either they are QTPP process, preliminary, control, strategy. So it involved like that. So we need to be minimize this quality risk, this risk assessment. Like you can see that I also you can see the initial assessment for drug substance attribute. So it usually we mark with the help of these three colors. This high, low, and medium. This is the standard color marking in the documentation as well, product development reports. So it involved by the identifying three levels: low, medium, and high. So how this drug product CQS XYZ is associated with the drugs attributes of the drug substance ABCD. So they are, we need to be work the substance attributes and the drug product sequence the got and we identify initially, and we mark this is the level of risk. If it is the case of low level, there is no do not worried about that. If it is in case of medium, we need to be work on initial. If it is in case of high, so we need to be so initially we need to work on the R&D to minimizing the risk from high to low. Well, so we need to be identify the risk, and then we need to work on the subset of the some experiment, and then we can say this is the high risk has been initially was found, and then we rectified, and we have to control these parameter, either they are the CPs or CMS or any parameter, and then we drop down to the low levels of the risk. You can see risk management method as per this this you can also say that design, process, material, facilities, or manufacturing, distribution till to the product has been entire, we can say like complete product lifestyle. We need to work on the risk always. Now coming to like one more important this program, that is the Fishbone Ishikawa. This is also used in this risk management. So this is the Fishbone Ishikawa diagram. You can see that. So that is also for if we definitely we work on the QBD aspects, all QTPP, CMA, CQS, CP, all or this we identify, and we successfully prepared one batch, even though one exhibit batch we prepared. What happened in that if we are going on a second batch, exhibit batch, and suddenly we got the failure mode? It happened. If we work on that, then only, because a lot of factor, for complexity is involved. So after that, we need like risk. We need to, whatever the risk we made, that we need to work on, definitely that we need to review it. And after that, we need to, what happened in that in the industry, we constituted one committee. That committee will ask to each like a process, they can be reviewed the documents and that one. So they this in this Fishbone Ishikawa diagram, with the help of this one techniques Ishikawa, to identify the factor influencing like this, the what is like a CQA. So they will be, even though talk with the pupils, they are involved in that product development. What have their experience, their training skills, their process understanding, product understanding, so that we call like interview, we have to be constitute what the environment of that area. If we am preparing one using one tank, what the environment we need to be checked, like humidity or what the oxygen level, what the temperature is that at that day. So with the help of QA and QC people, we need to be work on that, and we check it if there is a might be changing into the that environmental. That's why like that it happens. That's why in that, or the second side you can work on the product side, like raw material. What the excipient is, the first the raw material we are using, it is tested into the plant for their CPM, CMS, and if it is tested, what is the any change into their parameter. Like for example, if I'm using when we executed all batches with the medium grade viscosity, and some person has checked this raw material and he used that lot of the high viscosity grade. So the product has been reached into high viscosity like that. So initially that we raw material, we again have to check it out in the documentation, what their solubility, concentration, related substance, physical, chemical, or the polymer like in a HLB or like that, depending on that one. So that we need to work on that. And then second side is like a product, we need to work on that. So for identifying the risk, where it why this we got it the failure. So the characterization, we need to work on that, and we have check all the specification, the range, and where it have been involved. So we identify like only the one parameter has been not meeting the compliance. So like for example, particle size. So we need to work on the specific particle size. Then process, manufacturing process, like I use the HPH. You can see sonication. It involves sonication. It involves high drying. Is the HPH, high pressure homogenized. So like I mentioned number of cycles. If we subject it for like a four cycle, but however it require change in the time. The four has been excluded, but time has been little bit changed. So here we need to be identified. Then we need to be check and verify the process related criticality. So with the help of that, we got to understand about the risk, as well as if why the product has been going into the failure mode. This is the one case study for like a lipid formulation analysis one. So for understand. Similarly, like for also like HPLC method development, that is for analytical. So nowadays in the each industry have been utilizing a process analytical technology tool for this HPLC and LCMS or GCMS. So they can also work on that if like in a, we are working on HPLC and HPLC method is after some time, like by changing the one scientist previously work, and after 1 year some other scientist will work, and suddenly got the failure. So we need to again work on this with the help of this fish one discover. Wherever we work on, we need to relook on materials, mobile phase, instruments, parameters, sample characteristics, and the preparation or the column characteristic. So you can see also like is the sample characteristic and the preparation. So where we work on like the sonication duration, how much time they PK value of the drug, selection of the solvent, which solvent they utilize, whatever the polarity of that polarization of the drug. So that we need to again relook and verify, and then we find it. So this is called like a fish funnel. So this is kind of a fish kind of a schematic structure in that. Similarly, like for extruded particles, you can see. So the extruded particle, over that the different steps unit operation involved. So like blending, extrusion, characterization. For example, in this extrusion, so what the temperature we have been what kind of the configuration of this screw is there, what the speed, time, dimension, rate, torque, like this. We need to be relook and then we learn. That's why this complex product development has been very critical for development perspective from the product development side. So this would be very helpful for understanding the product also. This is for like in a one diagram for weight granulated technology or drug delivery system. As I know, I spoke about the QRM, that is the process risk assessment in the Q9 category. So here you can see also once we start the process product development, so we need to be work on the risk assessment, and then we need to identify the risk, and then we need to be analyze and evaluate, and we mark with the correlation but to the three different colors and mark as a low, medium and high. If they belong to the red color, we need to be work on the control strategies, then they would be coming to the medium or and the up to low level. So we by involvement of the process development, control strategy development, and then for improvement of that process. So that is very important one. Nowadays in each complex product development, regulatory agencies asking about the QBD implication and then risk assessment initial level. So you can see here also what is this why this they mark with the three different low, medium and high one, and if those are fall into the high risk, so because they highly impacting onto the CQS or quality target product profile. So we need to be minimize the risk from high to low, and those already having the low risk identified, analyzed, so there is no problem for that. So this is the one template for also for marking of the this risk assessment. Like you can see the drug substance attributes, drug product CQS, like that one. So like you can see also like particle size distribution has been impacting onto the roll up to onto the dissolution profile. So we need to be marked as a high, and how we can be changed from high to lower one. So we need to be set some kind of experiment as a supporting one, and then we consider like that it doesn't work on the particle side. We said this much is particle side has been of the drug substance required for getting of the such dissolution apparatus that is in a within limit. So this is really this one. Okay, this is I don't think so. We can design space that I love. It's a multi-dimensional combination and interaction of the materials input as well as processor. So that is a one that within so the product should be within our design space with the help of controller that I already covered. This is not one. Okay, this I think I already covered this one, control strategy. So it involved into the quality attribute assessment, materials, drug product, or the process is even though including of the packaging material also. So we need to also work on this. Additionally, this one is packaging specification. We also setting what kind of the packing material is that. What even though nowadays extractable and leachable guideline is also implemented. So we need to work on the date showing the data of the extractable and leachable data. Okay, this I think already covered. This yeah, I want to like just additionally I'm giving this one more like dissolution studies. It's like yeah. So nowadays a lot of dissolution apparatus with we can for innovate new product development was like complex or the nanosite. So as we know that US apart from some pharmacopoeial and non-pharmacopoeial dissolution apparatus is with us, and we utilizing out of most widely used USP dissolution type 1 and type 2 we usually consider and utilizing in wide way. Apart from that, also for nanotechnology, you can see like a microfluidizer we can use in that the dialysis membrane with the help of syringe we fill the product and then we subject it for the dissolution media, and then we check out the release profile. Like you can see a floatalizer with the different molecular weight cutoff. So this is kind of minimizing of more error. Here, like it is USB resolution type 4, that is the automatic one also. So that one we can utilize for the complex product development. One more technique, that is the microdialysis. You can see here at the bottom that I work. These are all apparatus where I work on the one US FDA product. In that, they want to be develop one in vitro drug release guidance for the nanotechnology. So they want to be developed by utilizing all techniques, and which one is the robust and sensitive one, so that we need to be submit to the advisory to the FDA. So in microdialysis, we can utilize the micron, very lesser amount of the sample, and then we check out with the help of LCMS. This is the one more flow understanding about that. This is, you can see the my adapter and the connector with that in the left side. And after that, we can subject it into the dilution with the help of peristaltic pump, and we set the certain limit, and then we check out the inlet and out this outlet, and then collected the minimum sample, that is 10 microliter like that, or like 20 microliter, and then we analyze with the help of, if the amount is higher, HPLC, and then lower amount, then we can utilize the LCMS technique also. Like, so design of experiment, we know about that. So in the concluding one, I would like to say that the QBD is very important for intended use to enhancing of the understanding about the process knowledge, as well as for product understanding, process understanding, so that there is a we can build the quality on the system, and then which we can as per the like regulatory expectation. So there is a minimum chance for failure of the getting the approval of the product. So that is the one. The QBD suite of documents we can understand, and we can say that this with the help of the QBD, or this all understanding, or the documentation risk assessment, that we can have get to the product into the market. This is brief about my books. Too recently we published one book in dendrimer and nanomedicine. Well, second one is micro and nanotechnology based product development. Over in micro and nanotechnology based product, I have one chapter on this QBD approach for this nanotechnology in a more exhaustive way. This is my current research team with the PhD scholar and the master student. Now I would like to thank to Annamacharya College of Pharmacy for giving wonderful and fantastic this invited talk to, so that I believe that I share the knowledge about that I know, and based on knowledge experience, and that I gained from the industry, and then I get from to the regulatory that one project from the USFD. So might be useful for you all for better understanding. I would like to thank to our NIPER, Ministry of Chemical and Fertilizer, Department of Pharmaceutical Science, and thank you Dr. Surya Prakash Reddy and all faculties who are involved in this training program sponsored by the ICTs. I would be happy if somebody question has been, so that I can share from.
Thank you, sir. Thank you so much for your valuable presentation on this QBD approaches, the development of many complex products.
Mhm.
And so definitely this information will be helpful for the researchers to apply of all these principles while they're carrying out their research. And so the participants, so you can unmute yourself and ask if you have any questions. So you can ask.
You're not audible. The
Naveen
voice
sir.
is fluctuating.
Naveen sir, can you tell me one more time?
Yeah, voice is fluctuating.
Yeah, okay. Okay, so sorry for that. And so definitely whatever the information you are sharing the participants, so will be definitely be helpful while they are carrying out their research work. And also I request the participants, so to unmute themselves and so they can ask the queries. So I request the participants, if you are having any queries, so please unmute yourself and you can ask.
Okay, sir. As a token of gratitude, we are presenting an e-certificate, sir. Nilesh, sir.
Okay,
This is a
thank
token
you.
of certificate, e-certificate, sir. As early, today evening, we will send you through the mail or WhatsApp, sir.
Thank you.
Sir, thank you, sir. Thank you for the wonderful session, sir.
Thank you.
Thank you, Nilesh ji, for being with
Thank
us
you so
and
much.
sharing your knowledge.
Thank you so much for that. I believe that I shared on this now my knowledge on the QBD and approaches might be useful for all whoever the participant they listen
Definitely.
patiently.
It's an excellent talk. Every participant will get benefited.
Thank you so much for that. Thank you. Have a good day to all for successful this AICT.
Thank you, sir. Thank you so much, sir.
Stay safe.
Thank you so much.
Sir, next. Sir, చూడండి.
Deepa madam,
దీపా మేడం.
please unmute yourself. Deepa ma'am.
సార్ సార్ జాయిన్ అవుతారు. మేడం సుజాత మేడం జాయిన్ అవుతారు సార్.
ఆల్రెడీ సుజాత మేడం ఉన్నారు అనుకుంటాను మేడం.
అట్లయితే నవీన్ సరే సార్ ఇంట్రొడ్యూస్. నవీన్ సార్ గో హెడ్ సార్.
మేడం నేను చెక్ చేస్తాను, ఒక నిమిషం ఉండండి. సుజాత మేడం ఉన్నారు మేడం.
నేను ఉన్నానండి.
గుడ్ మార్నింగ్ మేడం. గుడ్ మార్నింగ్ మేడం.
మార్నింగ్ మేడం.
గుడ్ మార్నింగ్
నవీన్ సార్.
మేడం వెల్కమ్ టు ద STTP
Yes, ma'am.
program, madam. Dr. N. Raghavendra Naveen will introduce you, madam. Naveen sir.
Yes, yes, sir. Thank you very much, sir. So once again, good morning to all the participants and speakers. So we are moving to the second session in the date year form. Speaker Sujata ma'am. Sujata ma'am is working as an assistant professor. Of pharmaceutical more than I should there. But more also, I'm currently India's as a principal investigator and the co-principal investigator. So from the two grants, so which are received from the DSP and the UGC. And ma'am has around more than 40 publications in a reputed national and the international journals, and also edited one book chapter. And currently, 3 PhD scholars are working under Sujata ma'am, and also attended so various conferences. And the ma'am received international travel grant, so that is sanctioned by the SCRB DST India to attend an international conference, so pharmacology 2014. So with this introduction, I request Sujata ma'am so to deliver the talk. Thank you, ma'am.
Thank you, Mr. Naveen, for the nice introduction. Am I audible?
Madam, you are audible, madam. I just make you a co-host, madam. You can operate
Yes.
the slide.
Before I proceed, I just want to ask whether the participants want to have a quick break of 5 minutes, because since they are in the session since 1.5 hours, if they want to have a break for 5 minutes, it's okay. Otherwise, I'll start.
Schedule from 11:30 to 1 o'clock.
Okay, fine.
Yeah,
If
madam.
the participants don't have any problem, I also don't have any
No,
problem.
actually, more number of the students, number of the participants are active up to 1 o'clock, 1 hour. But later on 1 hour, they are entirely moving.
That's fine.
Yeah, madam. Yeah, madam.
Before I start my topic, I thought let us have a quick break of 5 minutes so that once again they'll get all the energy back. We can have an interactive session then.
Okay, madam. Okay, madam. I just
Choice is yours. Choices of the organizers. If you want me to continue, I'll continue, or we can wait for 5 minutes.
no issues, madam. You can continue, madam. No problem.
Okay, then fine.
Madam, I just make you a co-host. You can operate your slides, madam.
Yes, yes.
Thank you. Thank you, madam.
Are my slides visible?
Ma'am, yes, ma'am.
Okay. Yeah. Good morning once again, all the participants.
Good morning, madam.
Today is the third day of your short-term training program. You might be listening about the QBD approaches since 2 days, a little bit different. I'm going to talk about a computational approach only, but that's a little bit different. Computational toxicology role in the product development. While introducing, you might have heard that I'm basically a pharmacology person. Pharmacology, everyone believes that we work mostly with animals. That's everyone's belief. Yes, we do work with animals. But this pandemic has taught us to explore ourselves and expand ourselves. Because of this pandemic, we are unable to work in our research facilities. So we have been trying to explore the possible approaches by which we can continue our research work, in which, fortunately enough, with the help of my colleague, Dr. Shaheen Begum, I came across this concept, computational toxicology. And we are taking baby steps in our department. I should say we are taking baby steps in computational toxicology. We are not well established in this area, but still, I think sharing this topic with the participants might definitely be added advantage to those faculty who are still pursuing their PhD or in the early stages of their career. So that since beginning of the research, they can adopt this new technology into their research. So quickly going on to my presentation, this is how it will be. The flow of the presentation has been divided into 3 sections. I'll first highlight why we need to work with computational toxicology, why it is important. I'm going to spend 10 minutes on that. Then I'll explain briefly about the methods that are used in developing computational technology tools that are available online. And my major focus will be on applications. If time permits, I like to give a short demo of a software tool where you can predict the toxicology of the compounds. Before I start my presentation, let me tell you, whatever images I have used in my presentation are not because of my own research work. I have copied them from various articles. I have properly cited them in the slides. So I hope there will not be any plagiarism issue. So this is what I'm going to speak about coming 1 hour or so. So let's begin with why we really require a computational toxicology. You might be talking with many of your friends. I hope in this team we have number of chemistry people, number of analysis people, a number of pharmacology people who do number of in silico docking studies. If you ask me, in silico docking studies have become overused nowadays. We don't know whether we are using it properly or not, but still everyone is using, even myself, I'm also using in silico tools. So I want to give a proper meaning of whatever tools we are using in today's presentation. This I have copied from an article from Science Direct, where they have beautifully explained the 4 stages of pharmaceutical product development. The same stages you can apply for drug development or also product development. Drug, in the new drug discovery and development also you have the same phases. Once the new drug is approved, in the product development also you have the same phases. You have pre-formulation studies, you have prototype development, scale up, biological aspects are involved, beginning from pre-formulation studies up to commercialization. On the left slide, you can see the involvement of different groups, the different teams of researchers that relentlessly put their efforts in the product development. I'm not talking about each and every team. I'm going to only talk about the biology aspect. The biology aspect is very much important in the pharmaceutical product development. People may ask, once drug has been approved, when it is entering into the market, why still will require biology aspects? we still require biology aspects. Even the drug is approved and entered into the market, you're working in form of different formulations. There also you require biology aspects. In biology, I'm not talking about pharmacological effects. I'm not talking about ADME effects. I'm only talking about the toxicological aspects, because these we generally miss. Once drug is approved, we generally don't focus on toxicological aspects much. We don't focus on toxicological aspects much. So I want to press, or I want to say that the toxicological aspects, we need to always watch in all the steps of product development. In all the steps of product development. When we say that toxicology as a part of biology, when we want to understand the toxic effects of the drug or the product, the metabolites, the analytes, impurities, whatever they may be, if we have to understand the toxicity, the one and only option we have, or we generally follow, or usage of animals. Usage of animals. Animal models are very well established and approved by all regulatory authorities. All regulatory authorities accept animal models. All regulatory authorities accept animal models. The reason behind that is the animal models has been well validated. They will give you the physiological information. They will give you the information in the living situation. Hence, they have been approved by the regulatory authorities easily. But let me show you this slide. You see the numbers. You might be wondering, what is this number? This is the number of usage of animals in United States alone in the year 2018. In the year 2018, United States alone, people has used more than 780,000 animals, with an increase of about 5% in a year, both in public and private sector, in which the 23% are the rodents, where they have mentioned as other covered species in the slide. Apart from other covered species, rodents, guinea pigs, rabbits, hamsters, dogs, pigs, all animal models have been used in the research. See the number. We are sacrificing more than 7 lakh animals in one country. This statistic speaks about only one country. Then what about the world? Think about the number of animals we are using for research in India. When compared to US, the number of researchers involved are more in India. Let us not talk about the output. Let us talk about the number of researchers involved are more in India. So definitely the number of animals used in research is more, in which majority of them are going for toxicological research. Majority of them has been going for toxicological research. Now the question, since 5 years after European Union has formulated the REACH guidelines, REACH, REACH guidelines, which makes it mandatory to reduce the number of animals in the product development. Already you have studied all the toxicokinetics, toxicology in animals. Why you want to use once again animals in product development? Even in the preclinical discovery, the regulatory authorities are causing lots of pressure on the researchers to think on, can we really replace animals in product development? You see the numbers, medical and veterinary R&D, 44% they are using. I'm not concentrating on the percentages of other areas. I concentrate only on the toxicology. For the toxicological studies, approximately 10% of animals are being utilized yearly basis. Whether it is industry or whether it is a research organization, on an average 10% of animals is used only for toxicity studies, not for exploring biology. Hence, the biggest question now ahead in front of researchers is how to reduce this and how to reorient our research protocols to implement this 5R principles. 5R principles, everyone will be aware of it. Everyone will be aware of it. This 5R principle says that we have to reduce the number of animals used in the research. We have to refine the procedures which we are using in animal research such that pain will be reduced to the animal. If possible, you should replace animal models. Whenever you work with animal models, you have to rehabilitate the animals until their natural death. You cannot sacrifice animals without proper justification. If possible, reuse the animals. So these are the 5R principles now, which is the basis for all preclinical research. Whenever you propose any protocol, the IAEC will first ask, what are the 5R principles? Have you implemented that? So this 5 are hanging on our head, and the question to reduce the toxicology 10% usage of animals, people has put ahead lots of efforts. The first efforts towards this direction is formation of an inter-organizational unit called as TOX21. TOX21 is probably the first kind of its organization where the researchers from different organizations have formed a group, and they have put forward the applications of in vitro models, in silico models, and when the regulatory authorities can accept them. So because of the efforts put by the TOX21 team, number of teams have come ahead. Now they are providing number of alternatives. Instead of using larger animals, we are using smaller animals. Instead of using smaller animals, we are using in vitro models. And now the situation is, instead of using in vitro models, we are using in silico models. So this is why we need computational toxicology models, where we are using in silico approaches to predict toxicity of the compound. But the biggest problem we come from the journey of in vivo to in vitro to in silico. In vivo are more acceptable because you are performing in the live situations. You have the three-dimensional structure, the physiological conditions are there, the disease you want to induce, you can induce, all those possibilities are there. But the biggest disadvantage is being the cost, time duration, and extrapolation of data between species. You're performing in rodents, you want extrapolated to humans, which is not practically possible all the times. When extrapolation is tough from animals to humans, how you will ensure extrapolation from in vitro studies to humans, in silico studies to humans? That is the biggest research gap still existing in the research in this area. Extrapolation from in vitro to humans, in silico to humans. For this, people has put forward a flow chart. This flow chart is very good. This is about before I go to that flow chart, please have a quick look of the finance involved, because finance is a driving factor for our research. So this is a Grandwire research organization. They said that this is a in vitro toxicology market share in the Asia Pacific region, not in the globe. In the Asia Pacific region only, you see pharmaceutical industry, we are using in vitro toxicological testing models more than 40%. Cosmetics are also using more than 40%. So put together the cosmetics and pharmaceutical industry, we have lots of scope for in vitro toxicological testing. So if we equip ourselves with these testing methods, which are accepted by the regulatory authorities, probably we ourselves can become independent researchers or entrepreneurs also. So this is a picture which motivates us really towards exploring the alternatives for animal research. As I said, now people are even move ahead of in vitro. Now they are going for in silico. The basis why they are going for in silico is accumulation of enormous amount of data in the research articles since 10 to 20 years, since 1 to 2 decades. Number of research articles utilizing this in vitro toxicological principles have been published in peer-reviewed journals, in scientific indexed journals. All these journals are filled of this data. So based upon this data, this is the workflow proposed by the computational biologists. As I mentioned, test chemical is always there. Whenever we talk about drug discovery development, product development, you always have a test chemical. Whenever we are in the process of development, you will have a range of assay methods. These assay methods, either you have in vitro assay methods, you have PBTK models, that is physiologically based toxicokinetic models. From all these models, you are getting lots of kinetic data. You're getting lots of kinetic data. For example, to put it very simply, let me tell you, a benzene molecule, a drug having a benzene ring structure or aromatic ring, when it is subjected to a in vitro assay, for example, a MTT assay, at what doses you are going to get the IC50 values. So you can expect the similar IC50 values for similarly structural compounds, or the compound having the similar physicochemical properties or similar mechanism of action. So there are a number of criteria by which you can expect the similar endpoint. Similar endpoint. So from the literature available from the in vitro assays, PBTK models, various parameters related to the chemicals has been collected and various databases has been put forward. Once the basis has been constructed, people have moved ahead to construct these two important events, that is toxicity pathways and AOP models. AOP is adverse outcome pathway model. AOP is adverse outcome pathway model. If I say that a drug is causing liver toxicity, the endpoint is I'm going to perform a LDH assay, leakage of the enzyme lactate dehydrogenase. So I know that this chemical, when added to a hepatocyte cell culture, how it is going to cause a membrane damage and how the LDH is released. So that pathway I know. Being a biologist, we know that is available in the literature. So all those points are picked and the pathway has been constructed. And in the same way, based upon this pathway, they have proposed a pathway, a model called as adverse outcome pathway. Briefly, an adverse outcome pathway will have an endpoint, defined endpoint, that is hepatotoxicity. What is the genetic change occurring? What is the cellular change occurring? What is the metabolic change occurring? Because of which endpoint is produced. That is the basic concept of a AOP. AOP, adverse outcome pathway. Endpoint, for example, we take hepatotoxicity. So what is the genetic reasons for causing hepatotoxicity? You will define in one column. What are the cellular changes that are occurring lead to hepatotoxicity? That will be another column. The third column will be biochemical changes. Endpoint will be hepatotoxicity. If you model in that way, you call it as AOP. So once the pathways and AOPs are clear, people have used the mathematical knowledge in here, the mathematical knowledge, the coding knowledge, algorithms, machine learning, artificial intelligence. Nowadays, we are speaking machine learning, artificial intelligence, but the computational biologists or the researchers, they are using it since a decade. Since a decade, these softwares, what I'm going to explain, they are not new. They are existing since a decade. So they have used all this computational knowledge, mathematical knowledge, they have mixed it with this biological aspects and they developed a model. They developed a model which they are calling as computational model. Which they're calling it as computational model. So from so simply to summarize this particular slide, what I can say from the literature, from the in vitro assays, in vivo assays, PBTK models, all the information, whatever we have published till now, that information has been gathered. They have developed a proper pathways, proper models. They have developed a mathematical relationships, algorithms, and computational models have been developed for them. Now you have to input your molecule, your new chemical or new drug, you will get output predicting the toxicity. Predicting the toxicity. So this is as simple as such. So let us focus only on these two aspects. Let us not bother about the homework done. This is not done in 1 year, 1 day or 1 year. It is output of more than a decade. OECD, US Environmental Protection Agency, number of international agencies has been behind this and they have been putting their efforts since a decade, because of which they were able to develop this kind of flow charts, workflows by which number of computational models are available nowadays. Number of computational models are available nowadays. So with this, I think I have established the basic need of computational toxicology and how In brief, the computational models are developed to predict toxicity. I'm not talking about mechanism of action. I'm not talking about the pharmacology. I'm focusing only on the toxicology, how they are going to predict the toxicology. Now, moving ahead. I have mentioned that we have computational models developed. We have computational models developed. To develop these computational models, they have used various approaches. Though I have mentioned the first three more important approaches, that is statistical based or called as QSAR models. Second one is expert rule based or expert structural alerts model. Third one is read across model and other approaches. Other approaches I have not highlighted, but I have kept in the summary slide. But these three models I'm going to focus a little bit more, because many of the computational models which you want to handle, they are functioning on these three kinds of methodologies. So let's talk about the first model, statistical based or QSAR model. Statistical based or QSAR model. So what is this QSAR model? Every one of us knows. Every one of us from our UG, PG, even our research, we know what is this QSAR. We are trying to quantify the activity we observed with that of the structure. But here, now the activity we are quantifying is the toxicology, toxicity, the response of toxicity produced in relation to the structure. To put it a little bit simpler way, let me tell you what is this, what they are exactly doing. We know that whenever I define a chemical or a drug, that is defined in terms of two-dimensional or three-dimensional properties, like physicochemical properties, log P values, partition coefficients, pKa values, molecular weight. Those are two-dimensional descriptors. Three-dimensional descriptors are TPSA, topological surface area. You have number of other three-dimensional descriptors also. From the in vitro data, whatever we have obtained, people has calculated the QSAR parameters of the drugs, and they have developed the mathematical relationships with the toxicity observed. Using that as a base, they have developed algorithms. For prediction of toxicity. So that is the basis of statistical based or QSAR approaches. When we are using only structural activity relationships, why it got the name statistical based? It got the name statistical based because whatever mathematical relationships you develop between the structure and the toxicity, that need to be statistically validated by using either regression model or correlation analysis. Any statistical method you have to use, and you have to establish the relationship. Hence, they are also called as statistical based models or QSAR models. Very popular models in computational toxicology predictions. The second one is expert rule based or expert or structural alerts, which is very, very simple. Without any medicinal chemistry knowledge also, we can handle this. Simple, very simple. From the in vitro data or in vivo data, when they get a toxicity response, they will try to study the structure of the compound and see that this particular presence of double bond O or sulfur, presence of nitrogen at that particular position is responsible for attacking the proteins, or is responsible for forming the DNA adducts, is responsible for causing the membrane damage, responsible for protein phosphorylation. So number of toxicity mechanisms, whatever we observe, are attributed to the structural features. So all this information you're putting in one together in one platform. If your new compound is having that kind of structural alert, it will predict that this compound might also probably will have the same toxicity pathway. So that is how the structural alert mechanisms will work. Moving on to the third one, read across. Read across is very popular. You're taking a source, you're taking a target, you're assessing, predicting the target, reading across the data from the source. I'm going to explain all these three with the help of few examples along with the other approaches. So whatever I have been talking till now about QSAR, for this I'll take an article in which published in Ecotoxicological QSARs. This is an article in which researcher Carr has explored the toxicity of 9 halogenated chemicals. These 9 halogenated chemicals, they are having 5 single compounds or 4 or tertiary mixtures. Means either a single compound or a mixture of compounds. He has taken both of them and collected the data, in vitro toxicology data performed in zebrafish embryos. So for this 9 halogenated chemicals, whatever he has collected the data, they have calculated the descriptors. To calculate descriptors, they use number of approaches in which a medicinal chemist will be having more knowledge. But this Carr group has followed the weighted approach in which they have calculated the 2-dimensional and 3-dimensional descriptors. Those descriptors, whatever they have calculated, they have formulated in the form of an equation. They say that so and so toxicity is because of this relationship. So this mathematical relationship they have developed in the form of a model using genetic algorithms. This is a machine learning program. Genetic algorithm is a machine learning program. Along with genetic algorithms, they have used the statistical tool of multiple linear regression analysis. So this is how they have developed the model. To validate this model, first they have trained using 16 compounds. They have taken structurally similar. So the model they have developed using 9 molecules, which is single and also a mixture. Then they have taken an external data set, which is not these 9, but having similar properties. They call it as a training set, from which they have predicted 2,324 data points, 2,324 data points they have predicted using this model. So 3 steps, simple. Model development, algorithm and statistics are included in the model development. You have to give a test or train set. Then you have to test using another set of compounds. Test set is external set. Train set is also external set. But train set is always having structural similarities with the compounds used for developing of the model. From this study, they very clearly mentioned that fluorine-based PFAs are having high toxicity when compared to bromine and chlorine-containing compounds. Fluorine-containing compounds are having high toxicity when compared to bromine and chlorine. So they have model development 9, train set 16, but prediction they have taken 2,324 data points they have predicted at a time, where they concluded that fluorine-based PFAs are having more toxicity. This is not any software. Let me tell you, this is not any software. These people, they have used the QSR model to predict the toxicity. We did not need all this knowledge for us to understand. Knowledge. If you ask me, I don't know what is genetic algorithm. No, I don't know. But I know how to use this QSR model to predict toxicity of my compound. That if we learn, that will be sufficient. But to understand how it works, so this is a little bit of basic information, how QSR models are developed. As I said, it is also called a statistical based method. I'll give you one more example in which this is how they have validated the relationship between log P and the toxicity. This regression analysis is very common in product development. Whenever we perform any activity, we try to perform the regression analysis, and we want always ask for value greater than 0.99. If you are in formulation development, if I am in vitro toxicology using cell lines, I will ensure 0.9. I cannot attain 0.99, but I ensure either it will be above 0.9. As all these kinds of methods are used, it is also called a statistical based method. But OECD makes sure that if you propose a QSR based algorithm, QSR based program, if you develop, OECD will accept it for regular clearance only when you have a defined endpoint The toxicity you have to define, whether it is mutagenicity, carcinogenicity, aquatic toxicity, ecotoxicity, that endpoint should be defined. Algorithm should be clear. There will not be any ambiguity in the algorithm. The statistics you have used should have high robustness value, and application should be there for whatever algorithm you have developed. And without mechanical or mechanistical interpretation, OECD will not clear your algorithm. Mechanistical interpretation should also be possible. Mechanistical interpretation means this log P is causing this toxicity. How? Whether it is because of effect on protein, effect on DNA, effect on any particular GPCR, that kind of mechanistic interpretation should also be possible. OECD has collected all such kind of QSR based programs and made a beautiful toolbox available for us. We need not break our heads with all this analysis. We just have to give our input and read the toxicology prediction. To get that tool, they have performed all this homework they have completed. So this is a little bit about statistical based or QSR models. Now moving on to the expert rules or structural alerts. So expert rules or structural alerts. Here you see, I'm not going to read the definition given by Dearden. Dearden has explained that structural alerts need not be only computer based. If manually also you are able to predict the structural alert, then also can be called as a toxicology prediction. That can also be called as a toxicology prediction. But for our ease, these structural alerts have been computerized in the form of an algorithm and is available. The examples here you see, this NH2, which is present on the aromatic ring, is responsible for interaction with the proteins, causing the toxicity with carbutamide. People has identified this structural alert not only in carbutamide, wherever your drug impurity, wherever, if aromatic ring is having free NH2, it is going to affect your protein. Whatever may be the condition, it is proved. In number of experiments has been proved. So the only option is either you have to remove your amine, like in the case of tolbutamide. Carbutamide is not in market. They have synthesized carbutamide earlier, but because of toxicity, they have converted into tolbutamide. If you identify that your impurity is having such kind of structural alert, it is mandatory that you design the in vitro toxicity method in such a way to prove that it is safe on proteins. So it will help you to design the step ahead experiments which is really needed for the regulatory clearance. Not only carbutamide example, I'll give you one more example. Primary amines. You see this aromatic amine, the earlier one is especially important because wherever it is present, it is converted into nitrinium, affecting your DNA. This leads to mutagenicity. That means whenever you have this amine, as you observed in the earlier carbutamide, or wherever you observe, you have to study for mutagenicity. Mandatory, you have to submit the mutagenicity assay. One more structural alert for our quick reference: alpha beta unsaturated aldehyde. Alpha beta unsaturated aldehyde is very, very important for skin irritation. It causes phototoxicity. It causes photoallergy. So if your drug or impurity is having this structural alert, the toxicology program will highlight those areas and will alert you that these may be responsible for the probable toxicities. If you say that you have designed the molecule or designed the product in such a way that the toxicity will not be observed, you have to prove it. If you are not giving the supporting toxicity studies to prove that they are safe, regulatory authority will not clear your compound. So this is the application of how the expert rule based or structural alert. This is only knowledge. Expert rule based is nothing but knowledge. We know that this functional group is responsible for so and so toxicity. So based upon our knowledge, we are developing the algorithm. Wherever the compound is having this functional group, that will be highlighted. that will be highlighted. Next is read across, which I mentioned, which is very, very important. You have a source molecule. You have a source molecule available in the literature. The source, complete information you have. The source, complete information you have, like its physicochemical properties, its pharmacokinetics, ADME properties, metabolism profile, mechanism of action, toxicity profile, everything is available and documented. Now your job is to predict the toxicity of the target. Job is to predict the toxicity of the target. So predict the toxicity of the target, you have to read across all the parameters of the source. All the parameters of the source, and then you have to predict that because of the similarities at so and so parameters, the toxicity might be predicted. The toxicity might be predicted. So this is how read across is done. This is how read across is done. Read across methodology is not only for toxicology. Read across is observed anywhere. Finance, business management, computers, where you have a number of research areas, read across methodology is available. But in toxicological programs, you're predicting the toxicity of the target by reading across the parameters of the source. Reading across the parameters of the source. So let me tell you briefly the important parameters which we are going to read across. Structure, molecular topology, functional aspects, physicochemical aspects, metabolic pathways, mechanisms of action, stress species, etc. These are very, very important parameters of the source. These parameters, by number of predictions, number of combinations of predictions, you're calculating the values, minimum, maximum, mode, median, etc. You're applying various algorithms, this Hamming, Euclidean, Cosine, these are the coding methods. These are the coding methods which will help you to read across the parameters with toxicity responses. Read across the parameters with toxicity responses. So this is a brief information about the three important strategies which are applied in the computational toxicological concepts. QSR models, structural alerts, read across. These three are very, very important. Apart from that, we also have other approaches, hybrid models. No software is using single model. They are using combinations. They are using combinations. When you use more than one methodology, you call it as hybrid models. People also use the normal in silico toxicity predictions like molecular dynamics, receptor binding studies to predict toxicology. In which very popular is off-target binding. Very popular is off-target binding. Whenever you are going for pharmacological profile, when you study that along with the receptor in which you are interested, it is binding to the other receptors, that itself indicates that there are chances of toxicity. There are chances of toxicity. So there are a number of other approaches, but their commercial application is less. Hence, I have not highlighted much. Only the three approaches which are very widely used or very wide in application I have mentioned. To summarize, whatever models are available, I'll use this chart given in Wiley Interdisciplinary Review of Computer Molecular Science. They have published it in 2016. This is a beautiful compilation of various methods with their advantages and disadvantages. Various advantages and disadvantages. As you go bottom of the triangle, the prediction is more accurate. But it has many disadvantages because getting those endpoints, getting those data is not always possible. PKBT modeling, pharmacokinetic, pharmacodynamic modeling, and their relationship with the doses, dosimetry calculations, they might not be possible every time for all the compounds. They might not be possible for every compound. So data available in this section is very less. But if we use these parameters to model or to develop prediction models, then they will be more accurate. In the center of the triangle, we have already discussed about QSR, read across structural alerts. To the tip of the triangle, you have UF model. UF is uncertainty factor model. uncertainty factor model. I'll not explain much about UF, but I want to tell only one point about UF. Uncertainty factor model is a model method in which you're trying to extrapolate the toxicity from one species to other species. From one species to other species based upon the dose. Based upon the dose. Whenever we report toxicity, we report in terms of NOAEL, BMD, RFD, reference doses, etc. So those doses you are trying to calculate, compare with the compound, and you are trying to extrapolate between species. So UF, uncertainty factor model, is having very limited application. As I mentioned, these three models are very widely used in various online computational toxicology tools available. So this is about the various methods that are used in computational toxicology models. So a little bit of theory until now. Now I will move on to the more of application part. This is the list of various computational toxicology tools freely available on net. You can download them, all of them, beginning from EPI suite to test. All of them are downloadable, user-friendly, freely available. You need not pay a single rupee for it. The methodology which has been used in each and every software has been mentioned in the table. For example, EPI suite, which we'll talk about, which is developed by US EPA, United States Environmental Protection Agency. They have developed. This is a statistical method which will predict only ecotoxicological effect of the drug. Ecotoxicological effect of the drug. You're working in an industry, you have effluents filled with chemicals, you want clearance from the environmental protection, you want to claim that you are not causing any damage to the environment. So this software will help us. You want to predict toxicity in the environment, you can use this EPI suite, especially for ecotoxicology. Oncologic, also by US EPA, which targets only on the carcinogenicity. Among all the models which are mentioned here, ToxTree, OECD are having more number of endpoints. You see the methodology they have used. They have used hybrid method. They have used statistical and knowledge base, means they have both QSAR, read across model, and they also have the structural alert method. They have incorporated structural alert, read across, and QSAR. All of them has been incorporated. So we call it as hybrid method. ToxTree is developed by EU, European Union, whereas OECD QSAR toolbox is developed by OECD. If you see the other models available, I have mentioned Caesar. I want to mention about Caesar. This is also a part of European Union Caesar project, for which it doesn't require any knowledge. It doesn't have any chemistry background. It doesn't have any pharmacology background. You should know what is the structure of your compound. That's it. If you know that, you can predict 5 kinds of toxicities using Caesar project model. Using Caesar software, you can predict 5 kinds of toxicities. Those are mutagenicity, which is predicted using Ames test, carcinogenicity, developmental neurotoxicity, skin sensitization, and bioconcentration factor. How much of the drug is going to accumulate in the body, that can also be predicted using this Caesar model. So this is the long list of the toxicology tools, freely available tools available in the net, which you people can try your hands on. Now I will move on to the commercial tools available, in which I hope many of you are aware of Simulation Plus. ADMET Predictor, this is a commercially available software which is developed by a pharmaceutical industry. They have developed the Simulation Plus, where they are predicting your ADME. They are predicting your ADME of your compound, along with the complete metabolic profile. This ADMET Predictor, which is available as Simulation Plus. TopCat is a product of Accelerase. This is commercial. They will never show you what algorithm they have used. They will never show you what algorithm they have used. In the free softwares I have mentioned, you can see the algorithm, what they have followed. But in this commercial tools, algorithms are not transparent. Algorithms are not transparent. The TopCat, the advantage of TopCat is it has clearly defined the QSR space they have used. They have used the optimal predictive space validation method. Optimal predictive space validation method. Medicinal chemists will have more knowledge of this. What is the space? Using this method, this QSR method, they have developed this TopCat software, which has number of toxicity endpoints. I want to mention about ACD Tox suite specifically. ACD Tox suite specifically, which is developed by ACD Labs in combination with pharma algorithms. Why I want to mention this? Because this is one commercial software which will predict your HERG toxicity. We call it as HERG assay. HERG. This is a potassium sensitive channel present on the myocardium responsible for toxicity, cardiotoxicity. We know many of the drugs has been withdrawn from the market, especially the selective COX-2 inhibitors. They have this HERG toxicity. So if you give your compound, you can predict whether it is having HERG toxicity or not. This ACD also gives you predict the estrogen receptor binding affinity, genotoxicity, CYP3A4 changes in the CYP3A4 levels. That is induction inhibition levels can also be predicted by using this ACD toxicity. So these are the few highlights of the softwares which are available. Let me tell you, this is not the complete information. This is the maximum possible information I have collected. There are few additional softwares available, but the most common has been listed in this earlier on this slide. So this is about the various commercial tools that we can use for prediction of computational toxicology. Moving on to the last part of my presentation, I want to show you how to predict toxicology of a compound using this computational softwares, for which I have selected one is LASAR, the other one is OECD QSR Toolbox. The reason why I have selected LASAR, I'll come back to this IAC protocol later. I want to go directly for applications. I have selected this LASAR because it is very, very easy. Very, very easy. This LASAR, the case you can open in the website given there. You click on it, you will open the LASAR website. I have selected ivermectin as a case study because everyone of us are going through pandemic. We know ivermectin is one of the common drugs used to treat COVID. So I have selected ivermectin as a test drug. If you open this LASAR, this will appear like this. LASAR in silico CH predict. This is the pop-up box you will observe. You have two options. Either you can draw the structure or you can copy the SMILES notation. My advice is always copy SMILES notation from PubChem. We have number of other sources also to copy the SMILES, but my suggestion is PubChem is always relevant, more relevant. So you have an option, either draw the structure or give your SMILES. Below that, it will appear like this. Select one or more endpoints. Acute toxicity, you can predict in Daphnia magna or fathead minnow. These are the two animal models in which you can predict acute toxicity. You can predict BBB penetration across the humans. Carcinogenicity, you can have mouse model. Rat model also. If you want to understand how you are going to predict using this particular organism, you have to click on the details of validation. If you click on the details or validation, this is how it will appear. This I have copied it for Daphnia magnus. The model they have used, the source of the model, The type of the statistics they have used is regression, training, compounds 546, algorithm, similarity algorithm, Tanimoto, written by Tanimoto, fingerprint and MP2D are the descriptors they have used. Fingerprint and MP2D are the descriptors they have used. You will have 10 to 15 pages data, which will include the IST protocol, IST protocol means in silico toxicology protocol. So if you click predict using that particular one, how you are going to predict? So that notes is given in this validate model. You can understand how the algorithm has been developed using this notes. If we are not interested how to develop this algorithm, let us just click what the Daphnia has selected. Then you can also select low observed adverse effect level, that is LOAEL. You can predict it in rat. Maximum recommended daily dose in humans, mutagenicity using the Salmonella typhimurium model. These are the various options available in LASAR. These are the only options available in LASAR. It is not having all the toxicity endpoints, but it is having the most important endpoints. So to work in LASAR, what you have to do? You should know the structure of smiles. Copy smiles, select the one what you require. Select the toxicity endpoint what you require. Bus, 2 steps. Then predict. If you click on predict, what will happen? If I click on predict, this is how the prediction will appear. I have selected only for acute toxicity and mutagenicity. So this is the prediction report, in which it says that ivermectin, there is no acute toxicity studies in Daphnia magna species, or it cannot find similar substances whose threshold is more than 0.2 mg per deciliter. Whereas mutagenicity, it is non-mutagenic. You see the prediction? It is non-mutagenic. The probability of prediction is 0.788. Confidence is similar to bioassay results. If you're performing a Salmonella bioassay in the lab, similar prediction you will be getting. So similar to the in vitro method, it has predicted that ivermectin is non-mutagenic. So in the process of product development, if you identify any of the impurity, you identify any leachable whose structure who you identified any extractable whose structure just give the structure or the smiles notation, predict whether it is having any toxicity or not. This is sufficient for research purpose, but regulatory authorities will not accept LASAR predictions. It will accept only OECD predictions. So OECD QSR toolbox, these predictions are accepted both by OECD, US FDA, US EPA, European Union, anywhere. They accept the predictions of QSR toolbox. If you predict the toxicity using QSR toolbox, you generate the QMRF report or prediction report, submit it to the regulatory authority. They will accept you and they will exempt you from the in vitro or in vivo that particular endpoint toxicity studies. The important features of QSR toolbox, the seminal features of the toolbox are: you can identify the structural alerts. You can predict the potential mechanism or mode of action, mode of action of toxicity, not pharmacology. And if possible, you can also fill in the data gaps. These are the important features of QSAR toolbox. If time permits, I want to give you a quick demo of QSAR toolbox. Organizers, it is possible?
Yes, madam.
Okay, then I'll stop my presentation here. and I'll try to share the screen. To download the QSR toolbox on your laptop, they have given an instruction manual how to download it. You have to download the server first, which will appear like this. This is how the toolbox server will appear. You have to download the server first. Once you download the server, then wait a minute, I have to open my toolbox. Just give me a second. You're able to see my screen?
Yes, madam. Yes, madam. Yes, madam.
Okay. So you have two options. You have to download the server. The other one is QSR Toolbox. The current version available is 4.4.1, launched in 2020. You can see both of them on my screen. It will take some time.
Madam.
Just a minute. I'm trying to share my screen. My desktop screen I want to share,
Okay,
but
madam.
I'm having some issues with that.
Okay, madam. No problem. No problem. No problem.
Is it visible now?
Yes, ma'am.
Yeah. So this is how the QSR toolbox will appear when you open. As I mentioned, download is little bit problematic. You have to follow the manual very clearly. You have to download two plugins. One is server, the other one is toolbox. The current version is 4.4.1, as I mentioned. You have 5 options. You have input, you have profiling, you have data, then you have categorization, followed by your report generation. So input, if you see, you have this various options available. You have this various options available. You can open a new file, you can close, you can save. I will directly go with input method. Input method, you have 4 options. One is CAS number, the other one is by using name. If you know the structure, you can draw the structure using this pop-up window, or you can give composition also, means you have mixtures. You're not having a single compound, you have mixtures. Then all the information can be given here. But I will go directly with name. You know what I'm going to give. I'm very much interested in ivermectin, so I will search for ivermectin. You see, in the database, you have having 12 constituents which are having the name ivermectin. the name ivermectin. Either you can select all the 12 or you can select one. I'm not selecting all the 12 because it will take more than 1 hour to profile. I'll select only one. So the point is you can input your chemical whose toxicity you want to predict either by using SAS number, name, structure, or mixture you can give. You can even input in the batch using in the form of smiles. Using the form of smiles, you can input in the form of batch also. That is also possible. So this is how you are going to input. If I click OK, you will get this. You will get this structure information, parameters, physicochemical properties, environmental fate and transport, ecotoxicological information, human health hazards. All of these endpoints you can study. You can get the compounds directly from any database already available or any inventory you have stored it somewhere. List, as I mentioned, in the form of text. Copy the smiles in all the form in the text, and you can upload here. You can upload here. And here itself, we can define the target endpoint. Here itself, we can define the target endpoint. If you define target endpoint here, then only you can be able to perform read across. Then only you'll be able to perform read across. But that will take lots of time. So I'm not going to use that option. So I have given input of ivermectin using the name format. Then I'll go for profiling. If I click profile, you see, you have number of profiling methods which will automatically select by itself. If you don't want anything, you can avoid it. The first one is predefined. The second one is general mechanistic. General mechanistic, where you can have biodegradation half-lives using various biovin methods. Using various biovin methods, you can predict estrogen receptor binding, half-life, ionization, amount of drug ionized at different pH, protein binding either by OASIS method or by OECD method. You might wondering what is this OASIS and what is this OECD? These are only the methodology they have defined. Only the methodology they have defined. So among all these parameters, you can select whatever you want. Whatever you want. You can even go for endpoint specific toxicity like acute oral toxicity, acute aquatic toxicity, carcinogenicity, dark skin. All this you can select. You also have empiric information. If you want, you can select that. This it will not be automatically selected. You have to select by yourself. You have to select by yourself. Toxicological repeated dose studies, if available, that can also be retrieved. So this information we can select. Profiling methods. These are the profilers. You want information for this particular compound. Whereas metabolisms or transformations, I have earlier selected this. That's why it is automatically selected. You can select the metabolism pathways also. Predict that what are the possible metabolites of this compound. Once you select profiling, it is not mandatory that you have to select all profilers. You can fix yourself to one aspect. Let me tell you, here more than 500 endpoints are given. If you sit comfortably and read, more than 500 endpoints are given. It's not mandatory that you have to read all the 500 endpoints. Select the endpoint in which you are targeting. Only select that one. After that, apply. Selected profiles will be applied to all the metabolites. Do you want to continue? Yes, I want to continue. You see, it is running. You can see the green box running over there, and you have got all the information here. Profiling is completed. Here itself, you can transfer it to the Excel sheet, or you can go to report and generate the Excel sheet. So this is how profiling is done. The next step is data. In data, what you can do, you can gather any missing endpoint from the profiling from the net. You're connected to web. Only in presence of web it will be working. So from web, if there is any other additional point, you can directly gather. By clicking gather, you can gather. Here it will ask you whether you want to gather all the endpoints, or it will ask you choose. I want to choose only human health hazards. No experimental data is available on chemicals of interest. So in the website, the additional data is not available because already the information has been profiled. You see here already the information has been profiled. Here, if you want to know information, any particular information, you have to right click on that. For example, biodegradation fragments, biowin method, aliphatic alcohol it is showing. You have to click on right click and you can see what is it, explain. It will explain you what is it. Or it will give the scheme. It will give the scheme by using which it has given that prediction. That profiling, how it has done, that information you can get by clicking right on that particular box or parameter. This is how the data is gathered. This is how the data is gathered. IUCLDI is a new feature in this one where you can get the database imported directly from another database. Directly from another database. You want to import data from TalkStream. You want to import data from Lazar. You can directly import it using this function. Then the fourth key available here is category definition, where you can define with metabolism, subcategories, combine clustering category elements. Always it is not, always it is not possible. Depending upon the chemical you have selected, depending upon the endpoint you have selected, these functions may work or may not work. So I will try to define it with metabolism. In terms of metabolism, I want to see whether there is observed rat in vivo metabolism available. Then I'll click. It will give me what I want to measure, whether it is parametric, profile, or similarity. If I say profile, it will ask me which profiler you want to measure. I'll say ECOSAR, aquatic toxicity classification by ECOSAR. Parent and metabolites of ivermectin, I want to once again go for profile. Profiler, once again, I'll select the same one because then only we'll have meaningful information. Then I'll say, okay. What it will do now? It will get all the metabolites, possible metabolites for ivermectin available. This may take some time. That's why I have not selected the 12 compounds earlier, because if I selected the 12 compounds earlier, what will happen? It will take, by the time I reach here, it will take 1 hour. So here it will ask once again what you want. The metabolic profile, I want for human health hazards. Is it available? No experimental data is available. Ivermectin metabolites are reported, but no human health hazards has been identified or reported till now. So that is how you can include the metabolites. That is how you can include the metabolites. So till now, what I have explained, how to input the chemical, how you can select the profilers, how you can select the data, and how you can define the category. Category definition is very, very important because you can include the metabolites using this category definition. Then the fourth step is data gap filling. Data gap filling, nowadays in this toolbar, in this version, you have an advantage. You just give the input. Directly go for data gap filling. You need not go for all this profiling, data category definition. You need not give anything. Directly go for data gap filling. Go for automated workflow. Automated workflow requires more than one. Standardized workflow, this is also, it will not allow to execute now. It is not possible to execute now because I have given profiling, data category definition, I have given already. So it will not go for automatic workflow. Otherwise, you can go for automatic workflow. Automatic workflow you will have only for ecotoxicity, whereas standardized workflow you have for 4 to 5 varieties. I suggest you try by yourself. First give input, directly go for data gap filling, go for direct automated workflow. If you click on the automated workflow, it will directly perform all the steps and give you directly the report and give you directly the report. Here, the other important technique you have to remember is you have to select trend analysis, read across, or QSAR. That depends upon your endpoint. This is where your knowledge will come into picture. Until then, it doesn't require any expertise. It doesn't require any knowledge. You should know only the endpoint. You should know the relevant profilers for that. If I say carcinogenicity, what are the relevant profilers? Ames mutagenicity is relevant profiler. Salmonella typhimurium test is another profiler. Micronucleus binding is another profiler. DNA binding is another profiler. So regarding that particular endpoint, I should have the idea of the profiler. That is sufficient. But for gap filling, I should know whether I have to go for trend analysis, I should go for read across, or I should go for QSAR. If I have group of molecules, I will go for QSAR. I'll go for QSAR. If I'm comparing it with the source compound, I'll go for read across. Or I want to compare it this particular compound with already available similar structural compounds, I'll go for trend analysis. I'll go for trend analysis. This now trend analysis is not possible only if you give directly input, directly come for data gap filling. If I have not given profiler, I have not gathered the data, then trend analysis was possible. Now it is not possible. So the workflow in QSAR toolbox is in 2 to 3 formats. One, input profiling data category definition report. Second, input data gap filling automated workflow. Third, input data gap filling, go for standardized workflow where it will ask whether you have to select a trend, read across or QSAR. Trend, read across or QSAR. This information you have to decide yourself. That's why they say that to perform QSAR toolbox, you should have the basic knowledge of the compound endpoint you are looking as a profiler, which method will applicable to assign your profiler, to attain or predict your profiler. After you complete this, this is how you get the data. This is how you get the data. You will get the prediction, you will get the data matrix, you will get the category classification in terms of metabolites, you get QMRF sheet. I mentioned QMRF sheet is possible only when you accept a QSAR model, and this QMRF report is accepted by all regulatory authorities. And you people will be definitely aware to perform a QSAR model. One molecule is not sufficient. You will take a group of molecules, then only you can perform QSAR, and then only you can generate QMRF. For this, we can generate data matrix report only because I have taken only one compound. If I select create report, it will take time and it will generate data matrix. And you can see the data matrix in this format. Excel sheet. You can save it in the form of CSV file or Excel file. You see, various information is given on the left side. Mechanistic, protein binding, acylation will occur. So definitely they bind with protein. Ionization at pH 4, there is no dissociation occurring at pH 4. Hydrolysis half-life as per Hydrogen, no value. Biodegradation, it will take months and longer. This biodegradation, they are talking in the environment. Toxic hazard classification by Kramer, high class. Toxicity, high, class 3. Kramer will classify toxicity of a compound in 5 classes, class 1 to class 5. So this belongs to class 3. If you see, protein binding, no alert is found. It occurs only acylation. Once again, ionization at 9, pH 9 has been given. DNA binding by oasis, no alert found. Biodegradation probability does not biodegrade fast because in environment it will be for a longer duration. So those were the endpoints we have selected. It has given. And I have asked for the toxicity. This I have already completed. You have to slowly check your Excel sheet, which will for you to pick the particular endpoint. You see, I want to show you how many profilers are available. I have not selected all of them. Protein binding alert for skin sensitization, no alert found. Acute aquatic toxicity classification as per Verhaar, class 5. It is not possible to classify. So it is not causing any aquatic toxicity. Eye irritation or corrosion rules exclusion. Aqueous solubility is less than so and so volume. So eye irritation is not possible. Metabolism half-life is slow. Reactivity is unspecified. Protein binding potency, lactone. It is having lactone in its structure. If you see the structure, if you enlarge the structure, the lactones will be highlighted in red. lactones will be highlighted in red, which is a structural alert, which is a structural alert for binding for proteins. For binding for proteins. Anyhow, it is not causing any eye irritation. It is not causing any skin irritation. Inclusion rules not met to predict that it causes skin toxicity, skin irritation. But the only problem is it is not carcinogenic. But micronucleus assay says that it is H acceptor, path 3 H acceptor. So chances of mutagenicity are there. Chances of mutagenicity are there. To exclude this, you have to plan a in vitro mutagenicity assay and submit the data along with this information. You need not repeat all the dermal toxicity studies, irritation toxicity studies, no required. This data is sufficient. This proves that it is not causing any eye toxicity or dermal toxicity. It only shows that there is a possibility of in vivo mutagenicity. So you have to perform that in vitro test and report that, supplement that, whether it is really showing toxicity or not. Whether it is really showing toxicity or not. So from this data, instead of carrying out 10 to 12 in vitro toxicity studies, you are reducing yourself to only 1 or 2. You're reducing only 1 or 2. So this is how you are going to read out the data matrix, which is produced by the OECD QSR toolbox. OECD QSR toolbox. If I go on moving, you will have number of parameters, as I have mentioned. The number of parameters, they are unlimited. 373 parameters they are giving. Profilers they are giving. If you have selected any prediction, they will say which prediction, which assay system based upon which the value should be given. Value should be given. So this is how we will get a report from the QSR toolbox. QSR toolbox. So I'll stop sharing this report. I don't want to save this. Cancel. So this is how you can use the QSR toolbox for the toxicity predictions. For the toxicity predictions. I'll wait a minute here. Any doubts, you can ask me.
Madam, sorry to interrupt in the middle. So we'll be having another 10 to 15 minutes to finish up the session.
Yeah. I'm going to wind up now. No problem.
Yeah. Okay. Thank you. Thank you.
So I hope QSR toolbox, though I have given a quick demo, it's very easy, user friendly. Maybe you will have a problem for the first time, second time, but you can easily access through those pop-up windows and you can predict what you want. You can predict what you want. So I'll stop sharing this QSR toolbox and I'll go back to my presentation. So I'll conclude the presentation with just one important slide. Whatever, is it visible?
Yeah, madam. Yes, ma'am.
Whatever software you use, this protocol you should be aware. This protocol you need not develop. But this protocol is already in the software, but you should be aware of this contents. Whenever you start your computational toxicology predictions, you should know which endpoint you are going to target. Your target is eye irritation, your target is mutagenicity, your target is developmental neurotoxicity. What is your target? That should be properly defined. And while mentioning the toxicity, that endpoint, you should also mention what is the need for that, why you are going for that. That should be clearly given. Then you have to select the suitable methodologies and models depending upon the data available with you. Now I have taken only one compound, ivermectin, because of which I was unable to perform read across and trend analysis. It is not required for a single compound. For a single compound, it is not really required to perform trend analysis or read across. Hence, I have not selected that. I have just gone for profilers and collected the information. That's what I have shown. Then, what is the experimental data they have included in the protocol that you have to go through? If I say Verhaer classification, how they have developed the Verhaer classification, you should have that basic idea. Then only you can select the suitable profiler and methodology. Then the toxicological effects or mechanisms, whatever should be included in the protocol, that information should be prior with you. This is not for us to develop, as I mentioned. Whenever you are proposing a new protocol, as I mentioned, OECD, you have only ecotoxicity as of now. Automated workflow, you have only ecotoxicity. If you want to propose toxicity as available in TopCast or ToxCast, if you want to include it in OECD, then you have to mention all this contents in the protocol. All this contents in the protocol, effects and mechanism assessment. Mechanisms and assessment and reliability scores are very, very important. Are very, very important. Here, I want to mention one important aspect, that whenever you select a parameter, it should be reliable, it should be relevant. When you combine both of them, then only you can get the confidence intervals. Then only you can get the confidence intervals, followed by report generation. A very quick view of how an IST protocol works out. You have the experimental data, statistical model result is also with you. You have attempted various statistical models, so output is ready with you. Expert alert result also you should collect, that is structural alerts you have to collect, read across results. All of them put together, you are giving an assessment score. You are giving an assessment score. One toxicity is not because of one mechanism. One toxicity is because of multiple mechanisms. For each and every mechanism, you have to give the assessment score. Assessment score. All of them put together will form the major endpoint. All of them put together will form the major endpoint. So this is a protocol that was involved in either the LAZAR, which I have shown to you, or in the OECD toolbox I have shown to you. That protocol they have implemented. What are the contents of the protocol? You can go and check anytime in the free toolwares, not in the commercial toolwares. So to conclude this session, the applications of IST, that is in silico toxicological predictions or computational toxicology predictions, these are the various areas which need to be explored where we have not still move ahead. Metabolite analysis, we can perform toxicity predictions of metabolites, impurities and degradation products, extractables and leachables. From this, you can develop a proper rationale or proper battery of in vitro and in vivo studies such that they can be submitted to the regulatory authority. Then only the regulatory studies, cost, time involved in the regulatory studies can be reduced. They can give you some exemptions only when all of this can be properly explored. I want to say one point here. Computational toxicology, we people are really very, very lacking. We have not really ventured into this area. Natural products are also people are applying, especially Chinese natural medicine. They have ventured into this and they have established a safety of all the constituents of Chinese traditional medicine, which we people have not done. Many of us in this group also, people will be working with natural products, but they have not used these tools for the natural products. So I hope this will be a useful tool for you people to implement in your day-to-day research. Thank you for giving me this opportunity. I'm open for queries.
Thank you very much, ma'am, for giving such a in-depth explanation of in silico and in vitro models in the product development, and also for such a nice demonstration of QSR toolbox. So for toxicology predictions. Now I request the participants so you can unmute yourself and you can ask the queries, please.
So I think I'll stop sharing my presentation.
Yeah, ma'am.
Sure, ma'am. Sure. So participants can please ask the queries.
Okay, madam. Thank you for your valuable session, madam. So, as a token of gratitude, we are presenting a e-certificate to you, madam. Later on, we will send you through the mail.
Thank you.
Madam, there are the number of requests, madam. Your contact details and also your this PPT, PowerPoint presentation.
Okay.
I already send you the mail ID to your personal chat box, madam.
Okay, I'll see that.
Okay, madam. Can you share? Madam, there is a question, madam. There is a question from.
Please.
1 second, madam. 1 second. So what are the, so there are some of the question, some of the delegates asking that question, limitations in REA.
RA. Can you please expand what is this REMA?
Read access, madam.
Read across.
Read across, yes, madam.
Okay. Yeah, the limitations with read across are collecting the data points. I'm audible? Yeah.
Yeah, madam. Yes, madam.
In the read across, the main problem which you observe is developing the protocol, because the number of parameters which you have to define will vary from the endpoint to endpoint.
నేను తిన్నానమ్మా ఒక అబద్ధాలు చెప్తున్నాను నేను తిన్నా టేస్ట్
Can I request the participants to mute? I'm hearing some background disturbance.
Yeah, madam. I'm entering muting. Madam,
Okay.
I think
I was telling about read across.
Yeah,
The main problem when I show the slide itself, you have number of points, data points you have to collect. Then only the read across will be validated. Otherwise, it's not easy to compare the target with the source. And the parameters which are included, will depend upon the endpoint. The parameters you have to focus will differ in carcinogenicity. The parameters you have to profile will differ in another DNT, developmental neurotoxicity. So if you have that sufficient in vitro data or in vivo data available, then only read across will be possible. And read across can be done for single compound or multiple compounds. That is about the limitations and advantages of read across.
Madam, there is one more question, madam. Is QSR toolbox is possible for multiple natural compounds formulations to predict toxicity of
Formulations is not possible. But individual chemical constants is possible, which we are also trying our hand with. We are taking a plant, we are collecting all the chemical constituents available in the plant, and we are trying to predict toxicities. We are first profiling the toxicities, then we will be getting a meaningful information, predictions, PMRF reports later on. That is possible, not for formulation.
Okay, madam. Thank you, madam. Thanks for your wonderful session.
Thank you.
So this is your certificate, madam. Wait, madam, one more. Okay, madam. Thank you, madam. Thank you very much, madam.
Thank you.
Madam, one more thing, madam. You need your mail ID. What are the contact details, madam?
Okay, fine. I have just copied that mail ID you have sent me in the chat box.
Okay.
I'll send all my details to that.
Okay, madam. Thank you, madam.
In the footer itself, you will have my mail ID, my contact number, and I'll share the PowerPoint of today's class also to you people. Thank you for giving me this opportunity, and I hope everyone is safe.
Yes, ma'am. Thank you very much.
Take care of yourself. Thank you.
Thanks, ma'am.
Thank you, ma'am. A very good afternoon to the respected speaker, Dr. D. Sujata Madam, and delegates present here. I need to mention my deep sense of appreciation to today's speaker who has enlightened our mind and shown us a new path. We thank you for being with us this pleasant morning. Thank you, ma'am. You have given the presentation wonderfully, and I liked your way of presentation. Thank you, ma'am. Thank you for being with us.
So much.
Have a nice day, ma'am.
Thank you.
I request the delegate. The afternoon session commences at 2 o'clock. I request you to be in time, and the attendance is mandatory. We look forward for the positive response and understanding. Thank you.
Thank you very much. Thank you very much to all. We will meet you by 2 o'clock. Thank you.